Hey, how's it going? Today we're going to be talking about mitosis. It's pretty interesting because mitosis is all about a way that we can make cells. So just to start us off, here's a little quote. Biology is the only subject in which multiplication is the same as division. This basically means when you want to multiply, you want more, you got to have cellular division. So today what we're going to be talking about is the cell cycle, things that the cell does to be able to grow and divide. If we look over here at this picture in the upper right-hand corner, we're going to be talking about all of this stuff, interphase, and of course mitosis. What does this stuff look like from here? And you might remember this from the DNA extraction lab. So how did this all begin? You started as a cell smaller than a period at the end of this sentence. Once upon a time, you were just a little egg hanging out in your mom's fallopian tube, and you were about the size of a pin. And now, take a look. So we grow, we're multicellular. That's a photo of me when I was in high school. I've grown a lot from a little, tiny little egg once upon a time. And same with you. So how did you get from like that? How did you get from one little tiny cell to you, your many multicellular you, awesome, amazing you? So we talked a little bit back, remember back in development, we talked about marullas and blastulas. And so how you do that is the fertilized eggs first start to divide, and divides, and divides, and continues. So you might have remembered, right, we have two cells, we start moving on, we have a marulla, which is a group of cells. And it continues and continues on and on and on until you're like 75 million cells. So what are some reasons for cells to divide? They can divide for reproduction, to make copies for asexual organisms. So these asexual, A meaning not, they have to divide because they only have one cell, for growth, for you to get bigger, and for repair and renewal. So every time I tell that story about you get cut, you want mitosis or meiosis. It's always mitosis. You don't want sperm and eggs coming out of your arm. We're replacing and renewing yourself. What does that look like? Amoebas are asexual, so they'll split off. I'll attach a video, and you'll be able to see that. And again, we have mitosis happening for development and to regenerate, right, to regrow. So this happens a lot in other organisms as well. If you cut the tail off, they'll regrow. Same as you scar up, you come back. So how do we make new cells? So taking a look at the cell, remember we have the nucleus and DNA. We talked about that last week, the double helix, this deal. And we have something called the cytoskeleton. So take a moment and think, what does the skeleton do in you? The skeleton supports you. So a skeleton in a cell, the cytoskeleton, allows you to have movement and to have structure within a cell. Okay? So again, cytoskeleton has to do with giving you structure, and that's why we have positive building right there. Continuing on, we'll talk a little bit about the nucleus. Inside the nucleus we have DNA, and that DNA is combined with histone proteins. I got a lot of people who were able to find some histone proteins when they were doing their DNA extraction. That's why you were able to see it. DNA is wound up really tightly, but it has a lot of proteins, so you'll see it. In the nucleus, it protects the DNA, and it stores it. So it stores it in this thing and allows things to come out through the pores. So we talked about transcription, and RNA can come out through the pores, but big things cannot come in and hurt your DNA. So what kind of molecules need to pass through? Think back to transcription and translation. What needs to come in and out? Just a quick check to make sure you know what's going on in the nucleus. What do you want to keep inside the nucleus? What do you want to let out? I'll talk a little about centrioles. What centrioles are they're a protein part that help organize where those chromosomes go. So you might see these things looking like this. They're like little stars. They shoot out, and they help grab the chromosome and allow it to pull apart. So that's what centrioles do. So how do we get the right things? You want to make sure that all your cells are identical, so you have to have an exact copy, right? So the A, T, C, G rules, you want to make sure you get all of that, plus important organelles and enzymes. Remember, enzymes are kind of important proteins that help you do what you need to do. If we take a look at this cell right here, we have some chromosomes that are staying orange. In the video I showed you, chromosomes are staying red. Notice we have the cytoskeleton along the sides as well. So what's a kangaroo rat? This is what this guy looks like. He's actually pretty closely related to an elephant, little tiny small animals that live out in the desert. So what goes on in mitosis? So mitosis, the way I like to remember, is P, not C. You'll see that you have a cell that starts, condenses the DNA, just like that video. We break down the nuclear membrane. From there, we have metaphase, we meet in the middle. Anaphase, we split apart. Telophase, we have two cells. So I'll teach you the mitosis dance, right? Prophase, metaphase, anaphase, telophase, cytokinesis. That's what happens in mitosis. So matching them up with what happens: interphase, prophase, prometaphase, metaphase, anaphase, telophase. Make sure you can say these and kind of match them up to what's going on. Cytokinesis is the actual pinching of the cytoplasm. You're actually breaking everything off. IPMAX. So what happens before you get there? There's something called interphase. Inter means between, so you're going between two phases. In interphase, this is your normal cell, just kind of hanging out, doing its job, and it's just waiting. So most of the time, when people look, they're like, oh, are my cells replicating right now? Are my cells replicating right now? I had a couple students say, I've never seen that before. Because most of the cells, your cells are just chilling. They're not really looking for anything to do. It has a life cycle, just like you have a life cycle. In the beginning, you're born, then eventually you say, hey, you know, maybe it's time to have kids, maybe it's time to go to high school. You have a life cycle, stuff you do. The same thing happens with cells. So first, the cell grows and matures and gets ready to divide. That's interphase. And eventually, in interphase, it does these things. G1 is called growth phase, gap phase 1. S phase would be kind of your, like, getting ready. You're replicating your DNA, the A, T, C, G, the crazy thing that looks like a dump truck, and what else did we say? You saw the two strips unwinding, and then you got mitosis. And that's what typically happens for most of your cells. Every once in a while, you have a cell that never divides, and they just rest, for example, nerve cells and muscle cells. However, people are seeing more research that we can actually switch cells back on to this growing and mature side. This is kind of a generalization. Generally, your brain cells do not continue to replicate throughout your life. They kind of reach a peak when you hit about 21. So I'm already on the downhill. They tend to reach a peak, and then you don't really replicate very much anymore. Interphase. So if we wanted to look at interphase a little bit more, interphase has three parts. G1, regular growth, S phase, where we copy our DNA, and G2, where we're doing just basically more growth. So you can keep in mind we have two growing phases, one copying phase. Interphase, we continue to prepare to divide, so we're basically taking care of our DNA. S phase, we're replicating, so A's, T's, C's, and G's. We divide up. Keep in mind that you have about 46 chromosomes. How much DNA is in a cell? It's about 3 meters, so that's about 9 feet. If I took it out, that tiny little strand, and held it, your genome is about 3 billion bases. So remember that video I showed you how fast it goes? It's pretty quick, and keep in mind mutations are errors. We don't want to get errors, we want to have a pure, clean copy. How do we organize DNA? We use something called a bead and string model. We can put things into chromosomes. So what we have is DNA as a long string, and we wrap it up in these things called histones, and that way we can condense it and pack it really tightly. So for example, right, when you have like long cords and strings, you want to wind them up. It's the same thing with DNA. We're going to wind them up. So looking at the levels, we have DNA, histones, chromatin, and the duplicated mitosis. So we package it all up. We condense it so it's nice and tightly folded. I already said all this, showing you more pictures. Here's what some chromosomes actually look like, right? They actually do look like little X's. When I draw them with you, they'll look like little flowers. I always draw mine, they look like little flowers. We have chromosomes. So what happens, we have something called a sister chromatid. The reason why they're called sisters is because they get copied. So we have 1, 2, 1, 2 sister chromatids. They're connected at the middle by a centromere. So right now we just got a lot of vocab. Sister chromatids get copied. They're homologous chromosomes, meaning they match up. So you get one copy from your mom, one copy from your dad. Homologous means the same. So we have copies, one from mom, one from your dad. A little more information. Alright, so now we're going to jump into actual mitosis. So what's actually happening in mitosis, some people call it the dance of the chromosomes. They meet in the middle and they split apart. If you ever learned line dancing, right? Do-si-do and do-si-partner, and they split up again. Again, 4 phases, just like I told you: prophase, metaphase, anaphase, telophase. What happens in prophase? We prepare to divide. So all this stuff basically means we're preparing to divide, we're getting ready, and so to do that, we're going to break down our nuclear membrane, we're going to have our centrioles move aside. Prophase, prepare to divide. Metaphase, we're going to meet in the middle, and we're going to get ready. So all the chromosomes line up, right? It's a dance, we line up in the middle. They meet at something called the metaphase plate, meta meaning middle, and then they'll get ready to separate. We talked about last time I talked to you, we talked a little about what happens if they don't meet. If they don't meet, you don't divide. So that's what happens in some cancer cells. What comes after metaphase? Anaphase! So anaphase is splitting apart, the sister chromatids separate, and what happens is they literally walk along these strings called the microtubules. They walk along to their sides. It's kind of like you split the room, or in the Bible, they split the ocean. Same thing. We're going to split these guys in half until the poles move farther and farther apart. We're stretching our cell out. We'll continue stretching our cell out until we have individual chromosomes. Until we get to telophase. You can see right here our nuclei are forming up again. And the spindle fibers disperse. So now we're trying to actually get away from each other. We want two separate cells. A lot of times I'll call them the two daughter cells. From there you get cytokinesis. You're splitting the cytoplasm. The pinching off. So if you've ever pinched like silly putty or pinched off bubbles, same thing. We're stretching out. What does this stuff look like in real life? Here's a whitefish blastula. So we got a fish, we got an animal, a eukaryote, just like you. More animal cells. And now plant cells. Very similar. It's the same across all kinds of organisms. Animal, plant, not bacteria. Very small organisms like amoeba will do this as well. A lot of times what we'll do is we'll take an onion root tip. So next time you go to the grocery store, chop an onion, take a look. You'll stain it. If you take biotech with me, or biotech 1 with Mr. Lay, or biotech 2, you'll actually do some of this stuff, or AP Bio, and you'll see all these cells, and you'll count them, and you'll realize how amazing it is that mitosis exists. How you control it, here's a couple reasons. The lesson is you want to control this thing. You don't want it just happening willy-nilly. You do want to control it at different parts. You have basically different stop points. Lastly, kinetochores. These are part of your microtubules that tell you where the DNA should align. And I want to hit again chromosome structure one last time to summarize it and wrap you up. We have the cell division cycle, all this stuff, interphase, cytokinesis, bringing us home. We have 3 different parts, right? Mitosis, interphase. Some books will say cytokinesis. So in summary, take a look at you. Can you answer this question? How did you get from there to here? About one little cell to many, many, many cells. It's only possible because of this thing called mitosis. Wrapping up, you have the stages: prophase, metaphase, anaphase, telophase, cytokinesis. One cell becomes 2. We got to copy the DNA during S-phase. So I hope that helps you out. If you have any questions, please let me know. Please message me. Please contact me. Do something. Don't just sit on your butt. Thank you. Have a lovely day, and I will see you later on the flip side. Take care.